Hi, in this short video, we'll talk about endospore staining. Now, many bacteria, especially that of Bacillus genus, forms endospore in harsh environments. And endospores are resistant to heat and harsh environmental situations, and they can survive in this situation for very long. And this particular endospore staining method actually determines bacteria that forms endospores. Because of their tough protein coat made up of keratin, spores are highly resistant to any type of staining method, so they won't take up gram staining. So the primary stain that is used for endospore staining is malachite green, which is highly penetrant. So at the end of the staining, what you would be seeing that these bacteria that has produced endospore would be stained in green, whereas the other vegetative bacteria that doesn't form endospore would appear something pinkish. In this video, it would be clear what is the basis of staining and why these kind of coloration is generated. So the step one is as usual smearing on the slide. You would take some amount of the culture and put it on the slide and smear it. Then in order to fix them on the slide, you can heat it quickly and briefly, and all of these bacteria would be now immobilized on this slide. It would be easier in staining procedure. Then we have to add the primary stain. In this case, the primary stain is malachite green. Alternatively, in some cases, people also use carbol-fuchsin, which is used for acid-fast staining. Anyway, the point of using carbol-fuchsin or malachite green is both of them are highly penetrant. And endospore wall is almost impermeable to anything. So in order to penetrate, we need to use a stain that is highly penetrant, such as carbol-fuchsin or malachite green. Anyway, carbol-fuchsin would easily penetrate in the cell walls or in this endospore wall. And once we add malachite green, we wait for some time for incubation such that the stain gets in. But honestly, this stain would not get in very easily. That's why sometimes we have to put these slides on steam. Steam would help these stain to get inside the endospore wall more efficiently. So we have to steam it. Then the step is decolorization. Again, we'll be using a decolorizing agent here to wash these stains. Now in this particular situation, the bacteria that didn't have endospore, they would not retain the stain. The decolorizing agent would actually wash away all those malachite green from those bacteria, and it is actually soluble in water, so it would be easily wiped out. But the endospore staining would be retained, because once the stain has entered that endospore wall, it's very hard to wipe that stain out and take it out. So while counterstaining with safranin, only the vegetative portion of the cells would be colored as pink or like reddish color. That is why we are going to see this characteristic appearance in our microscope. We are going to see the vegetative cells in red, and generally we are going to see this spherical staining of endospore